Hi everyone, this is the first part of the video on our final project that Joseph and I did on microarray k-means clustering analysis. So the reason that this question is biologically important is because we want to be able to determine differences between different cell populations. As many of us might know, all of the somatic cells in our body have the same genes in their genome, so we want to figure out what makes them different, and so we can measure the gene expression levels, which can also be called the gene signature for a cell. And I thought the figure on the right just gives a really simplified explanation. This would be before differentiation, and then cells go to different types and have neuron-specific genes, and there's also epithelial, and there's a bunch of different genes for different types of cells. So in research, it's really good to be able to test the differential gene expression. One of the older technologies we had was the Southern blot. The guy who made it, his last name was Southern. Related to that is northern blots, which can be for RNA, and that was a misnomer because the person who invented that last name was not northern. But anyways, the probing of the southern blot can measure different samples, as you can see. There's 3 different samples in the figure, but it can only do one molecular probe at a time. The probe is supposed to hybridize with specific genes, and if you have X-ray film development, that would mean that the gene was being expressed in that sample. And so another technology is microarray plates, and that's a newer technology that can detect RNA or DNA using hybridization and probes. So microarray technology can detect many more gene probes in a single assay, so it's much faster, it's much more specific, and it's way easier to reproduce. You can actually buy the chips that someone else had used in an experiment, and one of the disadvantages is that they can get expensive, and it isn't really as flexible because most people don't have the means to make their own DNA chips. But the basic process, you can see here 2 different cell populations, isolating RNA and getting complementary DNA from that, and then combining it with the microarray plate, which has gene targets printed onto it. So once the DNA is allowed to bind, each of these spots represents one gene, and the color represents the sample DNA that bound there. So green would be one sample, red would be the other, and yellow would be binding of both. And then dark spots would be genes that aren't expressed in the samples. Oops. And once the machine reads the fluorescence levels, it can be transformed into a gene expression matrix with genes on the y-axis and samples on the top row. And so this would just be numbers kind of corresponding to the fluorescence level. So once we have the numerical data, it's important, or this is a typical process that happens with microarray data, is that it needs to be normalized to make the data more reproducible and to account for like bad hybridization or specific, like maybe your fluorescence reader is different. So there's a lot of ways to do that. And so we actually used R in the RMA thing, which we'll get to later. Ooh, next, I guess. But then the ultimate goal is to find differentially expressed genes. So we decided to implement the k-means clustering algorithm to help do that. We didn't quite get to map those genes back to the annotation and summarizing it, but the tool would be helpful in finding potential targets. So the normalization is part of pre-processing, which is what I meant is really important. And Joe used the RMA-R package to do this. You can see we got the data from this database, and it was Affymetrix DNA data, which outputs the values in a .cel file. So RMA uses or takes in processes .cel files. So what happened in Joe's script is that the background correction happened, normalization, summarization, and what was returned was this tab-separated file, like we saw earlier with the genes in the first column and each sample on the top in the first row. So this is a scatter plot of the data points we just saw here. Actually, to get the coordinates, I combined samples with this A99, and that would be one of the sample sets, and so A samples were these three, and then not in the picture, there's three more, and that was the B samples. So that's kind of how the plot's set up. And this slope of 1, that's kind of linear, is what we would expect to see with good, well-normalized data with log 2 expression values. Not quite sure about this outlier, but I wanted to make it a little bit easier to understand the different gene expressions, so transformed our data into an MA plot, which is also known as a ratio intensity plot, and M stands for the log ratio, and A stands for the mean intensity. So the data is transformed on that kind of scale, which makes it kind of look like the scatter plot we just saw turned on 45 degrees to its side. M goes on the y-axis and A goes on the x-axis. And you can see genes located above 0 are upregulated and genes below 0 are downregulated. Most of the genes are kind of hanging around the 0 on the y-axis because, like we would expect, many of the genes won't change their expression. So this is our data once we transformed it using a formula. Basically, this is the mean intensity, and this is representing the ratio between the two samples. So our data actually looked pretty similar, except this looks like it was really upregulated, but I think those are probably noise in the data that we would want to fix somehow with our normalization. So this is the data that we're going to put through k-means clustering, which, just to review the algorithm, K is the number of clusters that the data will be put in, N is the number of data points being clustered, and N can be very large in this algorithm, and with microarray data. The result will be different with each run of the algorithm, which could be seen as a good thing if you are looking to discover new possibilities, or a bad thing if you're trying to reproduce the same result every time. But some of the issues that we could have is that the algorithm could get stuck in local optima, which would be kind of these. If we would have got a cluster stuck up here, that wouldn't accurately represent our data. Oops. Oh, sorry guys. Or we could choose a bad initial centroid, which would take a lot longer to readjust. So the idea is that first we initialize with k centroids, which can be chosen at random. We will assign each point to a cluster with the minimum Euclidean distance. Then we would pick a new data point based on average distances in each cluster, and then recluster the points. We repeat this step, I guess, until the average is below a specific threshold, so the centroids don't change. And when the centroids don't change, or no points are moved between them, the algorithm will break out of the loop. So these are some of the results we saw. This is k equals 2, and we were thinking that maybe we would have wanted clustered k equals 2 kind of in the other orientation, but this is how it happened. So you can see there's kind of a clear definition. There's not like any pink randomly over here in the red or any red over here in the pink. That could be seen as kind of a downfall of the algorithm because there's always a harsh separation between points. You can see that here too. And in real life, there's usually not such a harsh separation. A lot of things are kind of interconnected. But as we expected, our data was kind of separated, so at least the algorithm probably worked out. This was kind of a weird result because I used k equals 8 and my program printed out 1, 2, 3, 4, 5, 6, 7, 8, 9 centroids, and I thought there was only 7 colors when I made the PowerPoint, but I just noticed these 3 red spots, so I'm not sure if this is a problem with the colors or with the clustering, but something's not right. But it did work with up to k equals 6 or 7. Just once we got to 8,